Today we are joined by Dr. Karun Singh, Senior Scientist at the Krembil Research Institute, University Health Network, and Associate Professor, Department of Ophthalmology and Vision Sciences at the University of Toronto. His work focuses on neurodevelopmental disorders like autism spectrum disorders, as well as neurodegenerative conditions like Alzheimer's disease and Parkinson's disease, as well as vision disorders. Hi, Karun. Welcome to the show.
Hi, thanks for having me.
So here at the Stem Cell Network, we receive a lot of questions about Alzheimer's disease and Parkinson's disease, as well as other neurodegenerative disorders. Can you explain a little bit about how your work is helping to advance our understanding of these diseases, as well as other neurodegenerative and neurodevelopmental disorders?
Absolutely. So the work that we do in my lab really is focused on trying to study neurological disorders, but in a dish. And one of the issues has always been with any kind of brain disorder, whether it's developmental or an age-related disorder, is that we can't access the brain, right? A living brain cannot access it. There's very few ways of doing that. And so what we can do is use stem cell technology, where we can take a blood sample from anybody, especially somebody with a neurological disorder, and we can take that blood and turn them into stem cells in a dish. And then from those stem cells, we can take those cells and turn them into three-dimensional tissues that resemble brain tissue. So basically we can make brain tissue in a dish from that individual with a disease. Our work is really focused on taking these three-dimensional tissues that we call organoids, and these organoids can be used to study how the disease develops from anybody with a brain disorder. And what's most exciting is that without touching that person's brain, we can actually test different drugs and therapies in a dish without, having to go back to the person. And so this is a really exciting time in neuroscience because we can now basically create three-dimensional brain tissue in a dish from anybody. So really thinking about personalized medicine as a potential option in the future.
Excellent. That's really exciting. As we look to the future, how do you see your research influencing treatments? And are there any breakthroughs on the horizon, like cell or gene therapies, that could improve or reduce memory loss in conditions like Alzheimer's and Parkinson's, or better manage neurodegenerative conditions?
So in our lab, we really are focused on kind of two lines of research. One is we're trying to create different types of brain tissue in a dish. The technology has really advanced, and that's really exciting, where we can create different parts of the brain in a dish, or we can even create nerve cells outside of the brain. So, you can have nerve damage. We can create these cells in our lab, and we're trying to use them potentially for cell therapy-based approaches, so that if somebody has nerve damage or has a disease where nerves are destroyed, we can do cell transplantation to try to replenish those cells and grow those nerves and get that person back to being healthy. So that's one line of work that we're doing in the lab. The other is where you don't have cellular loss, but you have genetic conditions where you have, for example, brain circuit dysfunction. And so there the cells are intact, but they're not really talking properly to each other, right? The connections are not correct. And that's because of the genetic disorder, right? There's some genetic mutation that's present. So using gene therapies, what we're able to do is test out different gene therapies in a dish on that person's kind of brain circuits in the dish. And we can do all that in the lab without having to go back to the person. And we can kind of do this on a population scale. So different brain development disorders, for example, in our lab that we study that are caused by a genetic mutation, we can actually develop gene therapies that will go and fix that genetic mutation in the brain tissue in a dish. And then if that is successful, then we can maybe eventually go into clinical trials in the future.
Well, okay, thank you for that. As a leader in your field, what is the biggest challenge or barrier to translating discoveries in the lab into real-world treatments?
So as you can imagine, the brain is this enigmatic thing. We still don't know very much about it. And that is still the thing that is preventing us from really going full force into new therapies and going to clinical trials. So that's the biggest kind of impediment that we have, is that we still don't know enough about the brain as much as we study it. But what I'm really excited about is this technologies that are coming down the pipeline, that are in our labs now and even coming into the future, are going to be able to really understand how brain disease occurs at a very high resolution level. And that's using whether it's human brain tissue or tissue that we generate in the lab from a specific person with a neurological disorder. We are going to be able to understand their disease much better in the next 5 to 10 years because of the new technologies that are coming out. And when we have that information, we can develop therapies for Alzheimer's, for Parkinson's, and even neurodevelopmental disorders, because we have much more information and we know exactly the molecules or the brain cells that are dysfunctional, and we can correct that. So really it's a lack of information that's driving this, but we're really excited about the tools that are on the horizon.
Great. Shifting focus now, there's a common misconception that scientists spend all day sort of toiling away in the lab looking down microscopes. Could you give us a glimpse of what it's really like to be a scientist and what it's like running a lab, aside from conducting experiments?
So really, I see myself as a team leader, and we get together every day, every week. We try to make good decisions about the research projects that we're undertaking. So my job is really to kind of move projects forward and to always have communication with my team and what are the best decisions to move forward. So the other thing we do is research is very expensive. So we have to fundraise, and we do that is by writing grants. And so a lot of my time is spent writing grants to try to fund the very expensive research that we do in the lab. In addition, a lot of my time is trying to gather information about what is happening on the horizon and how can we bring that work and potential collaborations to our lab to be able to take the science that we have to the next level. So that's a lot of science communication and going to conferences, talking with other scientists, really trying to kind of educate others about what we're doing and bring in information about what others are doing outside in the rest of the world back to our lab, so that when we undertake the research that we want to do, that it's at the very highest level possible. And that way I feel like we're going to be moving forward faster when it comes to trying to develop new therapies, whether they're cell-based or gene-based for the future.
Thank you for that. My last question is, what attracted you into the field of neuroscience, and what's the most exciting aspect of your work?
What attracted me to neuroscience is the more we study it, the more we study the brain, we still don't know very much. And that still drives me every day. I want to know how our brains work, and more importantly, I really want to understand why we have Alzheimer's and Parkinson's and even neurodevelopmental disorders where you have deficits from a very, very young age. That really drives me because I really want to be able to develop therapies or help develop therapies for patients and families. I've had the good fortune of meeting multiple families with different disorders, and it really is touching to see how much of an impact that someone's disease has, not only on that individual, of course, but their whole family. And so what really drives me and what I try to drive home to the people in my lab is that the work that we do is not just to better science, but it is to develop and try to develop new therapies. And so that really is what drives me and what I'm most excited about as we move forward.
Excellent. Thank you. It's been so great having you on the show. To our viewers at home, you can find more episodes on the Stem Cell Network YouTube channel. Thanks for watching.